Hello, welcome to our lab. It's on enzymes, which is lab number 7, and we are group number 4.
In the experiment, we'll observe the effect of pH, temperature, and concentration on the rate of enzymatic activity using peroxidase. Data will be organized based on observance, and the conclusions will be made to determine the effect pH, temperature, and concentration.
Our hypothesis for this experiment is that we predict that the increasing the concentration of the enzyme will increase the rate of the reaction. We also believe that increasing temperature will also increase the rate of the reaction. If the enzyme is boiled, the enzyme will become denatured and the reaction will not occur. And if the enzyme is put in an acidic solution, it will become denatured and the reaction will not occur.
All procedures are followed according to the Enzymes Lab by Dr. Steiber, Bio 201, unless otherwise noted.
So background information for this experiment is that an enzyme is a type of protein that catalyzes a reaction by binding to the substrate and creating a reaction. Enzymes themselves are not used in the reaction and are able to be reused repeatedly. In the experiment, we will be oxidizing the chemical glycol using peroxidase as the reducing agent. And glycol is a colorless solution which will act as a substrate. When it is reduced, it becomes tetraglycol, which has a brown color to it.
In this picture, you can see our 5 solutions: buffer, peroxide, turnip extract, and glycol in its respective containers to avoid contamination. This picture depicts our solution, which were made by following the table, enzyme concentration mixing table. This picture displays the spectrometer being calibrated in order to test our solutions accurately.
The table displays the effect of enzyme concentrations on enzyme activity. As you can see, we have measured the absorbance in nanometers for tubes 2 and 3, 4 and 5, 6 and 7 in 20-second intervals.
The graph shows our data which was collected for observance at different concentrations. Tubes 2 and 3 express the linear line where the observance at each 20-second interval increased very slowly. Tubes 6 and 7 did not express a linear line and also had the fastest increased observance out of all 3 mixtures. Tubes 4 and 5 expressed the linear line the best out of all 3 mixtures. This was the concentration of enzyme we chose to use for our second procedure, which was the effect of enzyme activity with temperature change.
So the enzyme concentration explanation. In the previous graph, we plotted points from a 0.5 milliliter extract, a 1 milliliter extract, and a 2 milliliter extract solution. And then we determined which had the most linear line. In our conclusion, the plots from the 0.5 milliliter extract solution was determined to exhibit characteristics of a linear line, which determined the concentration of the turnip extract to be used for the temperature effect experiment.
The independent variable for our experiment will be time, while the dependent variable will be observance. The lines in the graph represent the rate of reaction as it progresses, or the change in observance as time progresses. Observance is measured in nanometers, while time is measured in seconds. The line representing the progress of the reaction with 0.5 milliliter of extract exhibited the most linear graph and thus was used to complete the temperature experiment. In the graph temperature versus rate, we are not expecting a linear line for points since each plot represents a result from experiment under different conditions, which means there is no direct relationship between them.
This picture shows our solutions which are being prepared for our test on the temperature change of enzyme activity. Here it shows solutions which were prepared earlier were then mixed with its counterpart tubes for 10 seconds. Then they were poured into a cuvette to be put into spectrometer for absorbance readings. Solutions were put into the cuvettes and into the spectrometer for testing, as this picture shows.
The table effect of temperature on enzyme activity shows the observance for each mixture of tubes.
Depicts a temperature reaction on enzyme activity. As you can see, the tubes which were in the 4 degrees Celsius steam bath had a steadily increasing absorbance. Tubes 6 and 7 and tubes 8 and 9's results had to be neglected due to the fact they were mixed incorrectly, which gave us incorrect results. Tubes which were in the 100 degrees Celsius steam bath had no enzyme activity due to the high heat denaturing the enzymes.
Using the slopes from the line from the effect of temperature on enzyme activity, we constructed this graph. The rate of reaction at varying temperatures. The temperature is the independent variable, whereas rate is independent variable.
Error analysis. When performing the effect of a temperature on enzyme activity, an error occurred which skewed our results. When we were supposed to mix test tube 6 and 7, we accidentally mixed test tube 6 and 8, and then when mixed test tube 7 and 9, which caused the reaction not to occur. Due to a limited amount of time, we were not able to complete the pH test on enzyme activity. All other experiments were followed by lab manual directions. Lab 7 enzymes provided by Dr. Steiber.
Our conclusion is that the hypothesis we made concerning concentration versus enzyme activity was supported during our experiment. Once the concentration was increased, so was the enzyme activity. When increasing temperature, the enzyme activity also increased. This was determined from two of our results. This is true up to a certain extent, since the result at 100 Celsius showed no reaction. When tubes 10 and 11 were heated in boiling water, and then mixed, the absorbance was measured and no reaction occurred.